Thank you very much for the kind invitation. It's a pleasure to be here. My name is Manuel Patarroyo. I'm a professor of adhesion biology. It's a relatively new discipline that studies the adhesion of cells to other cells and to the extracellular matrix, identify the adhesion molecules and the relevance of this process to various biological phenomena. I'm located at the Department of Microbiology and Tumor and Cell Biology in Solna, here at KI. I'm originally from Colombia, from a beautiful country in the northern part of South America. And there I studied medicine at the National University. And at the end of the 70s, I moved to Sweden, here to the Karolinska, to do research. And I had the fortune to have this couple of bright scientists, George and Eva Klein, as supervisors. And talking about inspiration, I could hardly find a couple that could be more inspiring than George and Eva. Not only because of the deep knowledge in science, but also because of their generosity and wisdom. I entered adhesion biology through the leukocytes, interestingly. When already in the first year of my PhD studies, I treated these blood lymphocytes with a very tiny substance and very small amounts of that substance called furball esters. And I observed that within a few minutes, the leukocytes changed the morphology, show these Europol-like structures that are characteristic of motile cells. And even more interesting, they start clumping, attaching one to another. This was then cell-to-cell adhesion. I got excited by this finding, and I thought it could have a biological relevance. So I decided to study it further. So I took these untreated lymphocytes, and I tricynized them on the cell surface, and then treat them with furball esters. I observed the morphological changes, but they couldn't adhere any longer. So this was the first indication that these cells had cell surface proteins that mediated adhesion, and I then decided to identify these structures. The leukocytes, they attach also to the vessel wall by interacting with the vascular endothelial cells. So it's not only adhesion between leukocytes, but also of leukocytes to other cell types. And this is a very important process in the inflammatory response, since the leukocyte has to attach to the vessel wall to extravasate. So the strategy to identify those cell surface proteins that mediated the adhesion was to test a number of monoclonal antibodies against cell surface proteins, trying to identify one that could block the furball ester-induced intercellular adhesion of the leukocytes. And after testing more than 200-300 antibodies, I found one called antibody 60.3. I obtained it from Patrick Betty in the States. And that antibody recognized what is now called the beta-2 integrins, or CD11, CD18 molecules. The antibody was recognized in the common beta chain of a number of heterodimers exclusively expressed by leukocytes. So this antibody allowed then the identification of this molecule. Of course, other groups in the United States and in Europe and Japan, they also characterize these structures. I did further characterization of these molecules together with Carl Gamber in Helsinki during postdoctoral period. I also contributed to the identification of a ligand of these integrins, namely the molecule known as ICAM1, that is expressed by activated endothelial cells. The attachment of the leukocytes to the vessel wall is extremely important, and it's one of the steps during the leukocyte extravasation process. Following adhesion, the leukocytes squeeze between the two endothelial cells and traverse the vessel wall and invade the tissues. And here is where they are going to defend us against infections by eating bacteria, but also they might destroy tissues in pathological inflammatory disorder, during pathological inflammation in diseases such as autoimmune disorders. So this finding that leukocytes recruitment required adhesion molecules, originated the idea that can we block these adhesion molecules and then use it as a novel treatment to for patients with autoimmune diseases or inflammatory diseases. And this was proven by a biotech company that produced a humanized antibody against one of these beta-2 integrins. The antibody is called efalizumab, and this is 2 patients with psoriasis. It's an inflammatory disease of the skin. The blood leukocytes infiltrate the skin and cause this pathology. Now, these are the 2 patients with psoriasis before and after treatment with the antibody against the adhesion molecule. So indeed, it could have a biological relevance, this finding I observed in vitro, and it had also a clinical relevance. The antibody is not currently used because it had some problems, but another antibody used against another integrin, namely the integrin alpha-4, has been extensively used to treat patients with multiple sclerosis, since that antibody inhibits the infiltration of blood lymphocytes into the brain and hence protect the patients or MS patients with the destruction that the lymphocytes made in the brain parenchyma. So leukocytes, they also interact with the extracellular matrix, not only cell-to-cell adhesion, but also cell matrix adhesion. Here I coat the Petri dish with the extracellular matrix protein, could be fibronectin, laminin, collagens. And I start focusing on cell matrix adhesion as well because the leukocytes, when they extravasate, they also encounter extracellular matrix proteins, as for example in the vessel wall here, where they encounter, for example, laminins, and also a number of fibers here made by collagen and other structures. In addition, I was finding a large number of similarities between the way how leukocytes invade tissues during inflammatory responses and another phenomena, how the tumor cells invade the tissues, also during various processes that will lead to metastasis. So then I shifted from inflammation to cancer biology and decided to analyze in more detail the molecular basis of the tumor invasion and metastasis and the role of these adhesion molecules, particularly of the extracellular matrix proteins. And I don't have to tell you how important it is to understand this process at the molecular level, since really nearly 90% of all cancer-related deaths are caused by metastases, and that soon nearly 10 million people die from cancer each year. So then I focus on the interaction of tumor cells with the extracellular matrix, and I focused on a family of extracellular matrix proteins known as laminins that I would like to introduce to you. It's because the tumor cells, they interact with laminins and other matrix proteins during several steps of the so-called metastatic cascade. The metastatic cascade results in the formation of secondary tumors originated from a primary tumors when the tumor cells, they detach and then they migrate. So during this migration, the tumor cells would interact with the extracellular matrix. Now they will intravasate and they will encounter also matrix proteins in the basement membrane of the vessel. They will travel to far away through blood or through lymph, and thereafter they will extravasate, again encountering matrix proteins. And here they will initiate a new colony of tumors. So at various steps of the metastatic cascade, the tumor cells are interacting with extracellular matrix proteins. And something that you will see at the end of my talk, the tumor cells themselves are producing extracellular matrix proteins. So I have focused on this family of extracellular matrix proteins known as laminins. Laminins are large heterotrimeric structures. They are composed by an alpha chain here that has a globular domain in the carboxy terminal region, a beta chain, and a gamma chain. the 3 chains are bound by disulfide bridges. Now, they are a major component of the basement membrane. Basement membrane is a specialized extracellular matrix that separates different tissue compartments. And they are huge molecules, and at present, as common in biology, we have complexity. We know of 5 different alpha chains, alpha 1, 2, 3, 4, 5, 4 different beta chains, beta 1, 2, 3, 4, and 3 different gamma chains, gamma 1, 2, 3. Now, by combination of heterotrimers, these 16 different chains constitute, at least 16 different laminin isoforms. I follow most closely the alpha chains because they display the most exquisite cell and tissue-specific expression, and in addition, they are recognized by different integrin receptors by binding, interacting with this region. So this is the laminin family. I usually address them through the alpha chain. I usually talk about alpha 1 laminins, alpha 2 laminins, alpha 3 laminins, alpha 4 laminins, alpha 5 laminins. I just recommend you to follow the relatively new nomenclature that describes the chain composition of the laminin. For example, the former laminin 9 is now called laminin 421. In other words, it's composed by alpha 4, beta 2, gamma 1. So the first number is the alpha chain, the second is the beta chain, and the third is the gamma chain. So that tells you the chain composition. The alpha chains have tissue-specific expression, and in general terms, we could say that the laminin alpha chains, alpha 1 and alpha 3, are expressed by epithelial cells, alpha 2 and alpha 4 are expressed by mesenchymal cells, and alpha 5 by both mesenchymal cells and epithelial cells. And their receptors interact with their globular domain. This picture from Miner et al. Shows the expression of the different laminin alpha chains in kidney, heart, and lung. Alpha 1, alpha 2, alpha 3, alpha 4, and alpha 5. As you see, the expression of each chain is very specific, and that's why we needed to go deep into each of these chains to be able to understand these molecules. These are indeed, these molecules are masters of tissue architecture. They are forming the architecture of the various tissues. Now, for example, keratinocytes, they usually use and produce alpha 3 laminins like 332, whereas the muscle cells, they usually produce alpha 2 laminins like laminin 211, whereas endothelial cells, they produce and use alpha 4 laminins like laminin 411. That's why I refer to these laminins also as the area code molecules, since they tell the different cell types where to be in the body. So what happens if we don't have a laminin chain, for example, a laminin alpha 2 chain? This is an inherited deficiency, genetic disease. Since alpha 2 laminin is expressed by muscles and nerves, so it gives origin to a disease called muscular dystrophy. But what about if instead of lacking laminin alpha 2, we lack the laminin alpha 3? We get a completely different disease. We get a skin disease. It's because alpha 3 is expressed by epithelium, keratinocytes. And this is a blistering disease. It's a terrible disease. The epidermis detaches from the dermis because the glue that is attaching the epidermis to the dermis is missing. The glue is alpha 3 laminin, the laminin 332. Then I have focused on the receptors as well, since cells interact with the laminins, and among the receptors, we have several families that interact with laminins. Maybe the most important family is integrins again. So in the integrin families, we have laminin binding integrins, but there are also members of the immunoglobulin superfamily that are also laminin binding. Among the integrins, we have in humans 24 different integrins. Each subfamily is defined by a common beta subunit. You might remember the beta 2 integrins I mentioned at the beginning of my talk. But concerning laminin receptors, the classical laminin binding integrins are alpha 3 beta 1, alpha-6 beta-1, alpha-7 beta-1, and alpha-6 beta-4. But in addition to integrins, other molecules such as members of the immunoglobulin superfamily, like this one called MCAM, or melanoma cell adhesion molecule CD146, are also laminin receptors. This is a specific receptor for alpha-4 laminins, for example. It's upregulated, strongly upregulated in melanoma. That's why it's named MCAM, melanoma cell adhesion molecule. That means that these sites are very interesting for the interaction of the laminins with the receptors. It could be an integrin or it could be a CAM. It would be interacting with the globular domain of the lamin. Another concept I would like to introduce to you briefly is the fact that cells, they can produce their own laminins and use them in an autocrine fashion. For example, this epithelial cell labeled with rhodamine was originally placed here and within a few hours has migrated in this direction by secreting, depositing, and using in an autocrine fashion the laminins. So these green dots are an alpha-3 laminin that the epithelial cell has used to migrate. So it's an autocrine, it's an endogenous laminin that is being used. If we block the laminins, we block the migration of the cells. Another concept I would like to mention concerning cancer biology is the plasticity of the tumor cells. Nearly 90% of all cancers in humans are of epithelial origin. They are carcinomas, breast, prostate, lung, colon, and so on. So it's very important to understand the physiology of the epithelial cells, and in this case, which laminins are used by these epithelial cells. So epithelial cells, as you know, they are polarized and they are interacting with the basement membrane that contains the laminins, and following a number of mutations that take some time to obtain, so the phenotype of the cells changes from a normal epithelium, goes to adenoma, then to carcinoma in situ. And if you look at here, there is an additional phenotypical change in these tumor cells during tumor progression. They lose their epithelial phenotype and they become more fibroblast-like, mesenchymal, a process that we call EMT, epithelial to mesenchymal transition. Now these cells become highly motile, highly migratory. So then we'll intravasate, then we'll transport it to distant sites, then we'll extravasate, and then we'll initiate a second colony, and interestingly, they will revert to the original epithelial phenotype by a process that we call MET, mesenchymal to epithelial transition. So is this plasticity that we have to understand, and this is important to keep in mind because laminins, the expression of the different isoforms it depends on the differentiation state of the cells. So I will give a few examples concerning the role of laminins in cancer biology. This is the first one. What about the alpha-3 laminins? Alpha-3 laminins, like laminin 332, alpha-3 beta-3 gamma-2, they are basically down-regulated in, for example, colorectal cancer, as described by a German group. Here in green is the alpha-3 laminin. This is a colorectal cancer. This is the center of the tumor. The basement membrane is preserved. But if you look at the invasive front of the tumor, the alpha-3 laminin is gone. It has been down-regulated. There is no basement membrane. There is loss of the basement membrane. And as you know, the loss of the basement membrane is strongly associated to formation of metastasis and the very short survival of the patient. So there has a clinical relevance. Interestingly, the alpha-3 and the basement membrane comes back when the metastasis has been formed. So here we have a classical example of a down-regulation of a laminin isoform of the alpha-3 during tumor progression. What about the alpha-4? Well, the alpha-4 behaves interestingly the other way around. We observed this together with the ladies Virtanen in Helsinki. We took oral epithelial cells from a benign tumor, and they, not surprisingly, they did not express alpha-4 laminins, since alpha-4 is mesenchymal, not epithelial. But when this tumor progresses and became malignant, then the malignant cells, they start expressing alpha-4 laminins. And they did it at the same time that they made the EMT conversion. They became mesenchymal. Now we also checked for the expression of the alpha-3 laminin, and it was exactly the opposite. The alpha-3 was expressed in the benign tumors, but was downregulated in the malignant tumors. So here we have an upregulation of the alpha-4 during tumor progression. I would like to illustrate also the role of the alpha-4 in tumor progression by describing a very malignant tumor of the primary tumor of the brain, the glioblastoma. It's a highly invasive tumor, and there is a Japanese work by Nagato et al. Has used a xenograph model in which they took glioblastoma cells labeled with rhodamine, human glioblastoma cells, and they injected them into the brain of immune deficient mouse. So after a few days, the glioma cells, they invaded the brain parenchyma in a quite characteristic way of the glioblastoma. Now, if they knocked down the glioma cell alpha-4 laminin, the cells were unable to invade. And this indicated that the endogenous, that the glioma on alpha-4 laminin was required for the invasion in the brain. A similar example have been described by Tabasoya in Rockefeller University by using a breast cancer model. As you know, breast cancer metastasizes to disseminates to the lymph nodes and to the lung and to the brain and the bone and other tissues. So by doing a selection system of, again, human cells, breast cancer cells, and then injected them into immune deficient mouse to produce tumors, you observed that those tumors in which you induce overexpression of the laminin alpha-4, so they readily form tumors and with metastasis. The other way around was also occurring. Namely, he took a highly tumorigenic metastatic clone of a human breast cancer cell line, and he knocked down the endogenous alpha-4 laminin, and those cells, they couldn't make any tumors any longer. So this demonstrates the relevance of these laminins in tumor invasion and metastasis. I refer to the alpha-3 laminins as tumor suppressor laminins, and the alpha-4 laminins as oncogenic laminins, oncolaminins. Mm-hmm. Now, this finding had also a clinical relevance, of course, because breast malignant lesions had a much higher expression of alpha-4 laminin compared to pre-malignant lesions described from different databases. So the malignants had more alpha-4 than the pre-malignant. And also, those patients whose breast tumors, had high expression of alpha-4 had much shorter survival compared to those patients whose tumors had low alpha-4. So there was a clinical implication also for this. What are all these laminins doing? Well, in the tumor cells, they are signaling through the receptors such as integrins and promoting, for example, proliferation, inducing invasion, inducing survival and other responses in the tumor cells. And that means that we have here a very interesting interaction that could be a therapeutic target to inhibit for example, the development of metastasis, to develop anti-metastatic therapy, for example, since these molecules, these laminins, are so important for metastasis. And this is undoubtedly a very interesting molecular interaction that could be a target, since receptors are recognizing the globular domain of the laminins. Mm-hmm. So in summary, I would like to tell you that laminins are extracellular matrix proteins, are mainly found in basement membranes, specialized extracellular matrix. They are masters of tissue architecture, and since the tissue architecture is rearranged during tumors, tumor metastasis, for example, it's not surprising that the laminins are altered in cancer. Now, the laminins isoforms, there are all nearly 16 different. They are expressed in cell and tissue-specific manner and are recognized by a number of receptors, including integrins and other molecules. Now, the tumor cells express different laminin isoforms depending on their differentiation stage. You remember this plasticity, and are able to use the laminins in an autocrine fashion, secreting them and using them for migration, for survival, and for doing other things. Now, during carcinomas, alpha-3 is usually downregulated, and that's responsible, at least in part, for the loss of the basement membrane, as described by pathologists, for example, whereas another laminin, the alpha-4, is upregulated and appears to contribute to the ability of the tumor cells to invade and metastasize. So laminins are promoting then tumor cell migration, tumor cell proliferation, tumor cell survival, but also stemness, the renewal of the tumor cells, and something I have not mentioned to you, the chemoresistance. They make, the laminins make the tumor cells resistant to therapy. Well, with this, I would like to end by acknowledgement that various people that have contributed to this group, to this work, members of my group. I have had a long-standing collaboration with Carl Trygvason. We have been characterizing these molecules for many years. My part has been to focus on the human laminins, isolation, purification, characterization, and also generation of reagents, particularly monoclonal antibody against these structures. And I've been also having a long-standing collaboration with the group of the ladies Virtanen in Helsinki, also here in Stockholm, Johan Hansson, melanoma expert, as well as Takako Sakaki in Germany, Arturo Mercurio, and Peter Yushchenko in the United States, and more recently with Science for Life Laboratory. With this, I would like to end my presentation, and I would be most glad to take questions. Thank you very much for your attention. Thank you.